Our lecture for today is about next step after sequencing to know HLA alleles by using Codon Express genotyping software. In this presentation, I am going to talk about my experience in using Codon Express for HLA typing and alignment to database. This method was a gift from my Canadian co-supervisor, Professor Dr. Malu, during my research scholarship in Manitoba University and College of Medicine in Winnipeg, Canada. Also, I am grateful to Trevor Peterson for HLA typing training, of course. Now, Codon Express is a genotyping software based on a taxonomy-based sequence analysis, TBSA, to resolve heterozygous combinations. Developed for sequence-based typing, SBT, of HLA genes or any MHC genes. I mean for any major histocompatibility complex genes. It's used to type any polymorphic gene with a database. Also, it can analyze chromatogram output from a wide selection of DNA automatic sequencers like ABI, CEQ, and others. It can also be applied to analyze sequence output text files. It's easy to use, flexible, having many functions that can assist user to resolve difficult sequences. Now, navigation the menus. The menu bar of Codon Express has 6 drop-down menus and many function buttons that allow user to access the different functions quickly. It's divided into 2 separate screens, allow user to view trace file, typing result at the same time. Also, user can choose display trace file, typing results, sequence evaluation, database in any one of the 2 windows by a simple click. Now the file menu. The file menu lets users to load the sequence database, load sequence trace file or text sequence file, load test, the typing results stored in the computer or on disk, choose the format of input sequences so that sequences from any source and in any naming can be loaded according to individual samples. Also, it allows users to start batch typing procedure, save edited trace file or typing result, export sequence file in FASTA format, also to set up print functions and print. Now, how to use the sample naming function. When you select sample naming function, you can activate sample naming submenu that allows you to define the name of your trace file or text file. Here, for example, a sample was sequenced with both forward and reverse primers for DPB1, B12, 10120, core DPB sequence F04, the forward primer here, and B12 10120 PID DPB sequence R04, the reverse primer. Here, the most important in the sample naming is the patient ID. For example, our patient sample here ID, the ID was DPB116. This is the ID of our patient, 16. patient number 16. The menu, the view menu. The view menu allows user to view sequence trace file, current typing results, sequence evaluation, codon assignment, loaded database, typing results. The typing menu. The typing menu, using typing menu will allows us to activate the table typing dialog, or we call sequence typing dialog, activate the trace editing dialog, activate evaluation sequences function, select reverse complement sequence, validate typing results, validate codon assignment, and find classified codon in that case. The typing table dialog, we also call it sequence typing dialog, consists of several functional buttons. Buttons allows user to check the typing results of each sample and to do manual corrections whenever necessary. The trace edit dialog. When we select the trace edit dialog, it activates a sequence trace edit menu. User can use it to edit sequence trace file by a simple click. Here you can see many buttons and each one have specific function in that case. The sequence evaluation. This submenu is activated when users select the evaluation sequences option. It allows users to select alleles in the database to compare with current typing result to find out the missed sequence call in the typing. Here, for example, the marked one, DPV1 020106. Here we can choose it and select this allele and choose the sequence evaluation button. The validate typing result dialog. This submenu is activated when there are multiple combinations in the typing results. Like here, we have 3 options. The first one, 030101, 1701. The second, 0601, 3501. The third, 2901, 9801. In this case, the validate typing results dialog, we can choose to give us the chance to choose the appropriate one after validation. We can choose, for example, the first one, 030101, 1701, and marked on it and say, then OK. Now the settings menu. Second menus. By using this menu, we can select or unselect the trace typing option, or we can select or unselect automatic typing. In that case, only the sequences with exact match, when we select, I mean the exact typing option, only the sequences with exact match with database will be displayed. In case there is no exact match, either due to a new sequence or the quality of the sequences is uneven or mixed, users can unselect this option and conduct sequence evaluation to find out the reason for the mismatch. That means we convert to automatic typing in that case. Automatic typing. As soon as the sequences are loaded in the typing program, an automatic typing is carried out and the result will be displayed in the View Current Typing Results submenu. Also, here users can unselect this option and return back to the exact typing according to their sequences, of course. Also, this setting menu allows user to select option for saving the edited trace file and select the font, select toolbars to display. Now the toolbars. Toolbars allows users to select or unselect different toolbars to display in the typing menu. As you can see here, many submenus: exact typing, base call read, exact typing, save trace excluding cutoff, display database in simplified form, list typing sequence, list codon data, choose font, toolbars, keyboard, mapping, and in the toolbar we can choose the main toolbar, test toolbar, trace, or scale toolbars. Their icons will be displayed in the typing menu and users can access their function by click on the icons. Now the base call read menus. Base call read menus include proofreading start, proofread faster, medium, or slower. The recommended one is proofread medium in that case. It's the recommended one. Now, before we start typing, let's mention the colors of the sequence in the chromatogram. As we can see here, the T with red color, the G black, the A is green, and the C is blue. In the upper two, we have homozygous GG. We can see here the peak of G. And in the second, we have homozygous, for example, here A. We have many homozygous A peak. Also, we have G also homozygous and T here in the second one. The second row, we have heterozygous peaks. In the first one, we have heterozygous G and T. You can see here the two peaks appear together, the T and G, black and red. Two peaks appear together, mixed. Here, that means the heterozygous. Here, G and T. In the second, we have two peaks also, heterozygous A and G. The green here and the black appear together mixed. And the lower one, the last one, the third row, we have homozygous T, TT, and we have, you can see here, we have 1, 2, 3, 4, 5 T here, and the second homozygous GG. We have nearly 5 peak of G. That's mean homozygous GG. This is a normal situation. When we are doing our typing, sometimes other letters appear, not A, C, T, or G, other one, like V, D, K, R, B, H, M. What's that mean, each one? When our sequence indicates these letters, like V indicates A or C or T, that's mean one of these three is our sequence. D indicates A or G, K, C or G, R, T or G, and B, A or C. The H, A or T, M, C or T. That's mean when we see these letters V, D, K, or either one of them, that's mean that there are one of the two base pair appear here. Now let us start our typing procedure. For example, we are going to type our sequence include DPB1 typing. The Codon Express can be used, as we said previously, to type any polymorphic genes as long as we have a database for that polymorphic gene. So we are going to compare, this software compare our sequence to the database and give us what are the type of our alleles in that case. Step 1. Load the DPB1 sequences. Select Load Database option or click the Load Database icon. Then select for the following example DPB 2004 sequence to open the database. As you can see here the arrow, we will select DPB 2004 sequence and then press open. Now the sequence database will be open. In that case, all the database will appear here. Now activate the sample naming from the file. We are going to go to the File menu and choose Sample Naming. It will be activated in that case. Step 2. Load your sequences. As we mentioned previously, we should let program know our samples name. We can load many sequences at a time by highlight all the sequences and click Open button. Like here, we highlighted all the sequence C12, D11, D12, E11, E12, and then click Open, and F11, F12, G11, G12, H11, all these will be highlighted, and then click Open button, and then it will be opened. Or we can select indicate one sample only, not all our sequences. One by one, we choose one by one. In that case, we can indicate one sample name only. Here we indicate 10112, and then in order to start the typing. Now, if the sample sequence, now the software will compare our sequence with the database sequence and give us the result directly. In case the sample sequences are good, then here is the result of our typing. Result typing will automatically displayed. Like here in this figure, DPB1 010101, DPB1 020102. This is our typing result appear automatically because here our sequences are good and need no typing procedure. It directly compared with database and the software give us our result. We can save the typing result and move to the next sample through the typing return to the typing table dialog. If the samples here marked with this between two brackets, small brackets, that means the typing is complete and the result is correct, as you can see here. It's very important to know the codon color map. If the codon appears it will appear in many colors, like this. If it appear in blue, dark blue, that's mean unchecked codon. Pink mean checked codon. If it appear in orange, that mean typing codon. In brown, that mean matched codon. In green, unmatched codon. In light blue, erased codon. And in gray, missing codon. Each codon will display in different colors according to the status of the typing of this codon. Now, if the sample appear without this mark between brackets, this indicates further examination of the sequence is required. We need to examine the sequence, examine the peaks or in chromatogram, and see if there is something wrong in our sequence. You can see here in the typing dialog, some codon are in brown color, like these, brown color, TTC, TTCGAC, CAG, many brown colors, codons in brown colors, indicating that these codons have been used for the typing and matched database in that case. But if there are codons in green color, this indicates that they are unmatched and need to be examined carefully, like this here. We have GCG. This appears in a green color, and we have to examine it carefully in order to let the software type these codons and display the result carefully. Now, how can we examine the unmatched green codons? We click W button in the typing dialog. This will sort the green codons to the top of the window, and then click on the green codons. The program will bring the sequence trace to the trace window. Now, as you can see here, We have the sequence and we have to examine each one and follow each sequence and compare it with the database. After confirming all the green codons actually exist in the sequence trace file and are not the miscalled traces or background, we can conduct an evaluation to find out whether there is a low sequence trace missed or it's a new allele. Sometimes a new allele appears during our typing. We can record that we have found a new allele in that case, or sometimes there are many missed calls in that case, and this is the reason why our typing appear incomplete typing. Now press evaluate in order to see what's the problem. When we examine the sequence trace to see if there's any missed call, and we didn't find missed call, in this case, as you can see here, codon 91 appear in green, and we have 1, 2, 3, 4 to 5 codon. These are mismatched, and 91, 92, 93 here mismatched. In this case, codon 91, for example, at the top of the codon testing trace, and there is no missed call. When we found these peaks, we checked one by one all the peaks here, and we found no missed codon here in this case, because in the DBB1 database, not all the alleles have complete sequences at exon 2. This is the reason why our typing result appear incomplete typing. So the next step to complete the typing, we are going to remove these codons manually, codon 5 and codon 91. Press on the green codon while pressing Shift key and see if this is the case. We omit this, delete this codon, remove it from from our sequence. Once the codon 5 and codon 91 are removed and selected, then click the typing button of the codon testing editor. A typing result will appear and indicate if the typing is complete. That means DBB1 1301 does not have sequence at codon 91 and do not have exon 1 sequence in the database. Here you can see here our typing is complete now. Typing result our sequence indicate that it's DBB1 030101 and DBB1 1301. This is our typing result. Now it is complete result. Thank you very much.